It's a pleasure to welcome Sue Washer, the CEO and President of AGTC, to share a little bit of the story.
Well, thank you, Josh. And Josh promised me this would be a little bit less formal, so I'm only going to show a few slides, and then we're going to have an interactive discussion. Just a few of the company highlights. We are an orphan ophthalmology company at our base. We have a track record of progress in that we've brought 3 different programs from early preclinical development all the way into human clinical trials. We have extensive expertise. We've been a company since 2000. 5 of the world leaders in AAV gene therapy were our founders, and we have an extensive platform of intellectual property and a broad pipeline. So why ophthalmology? You know, from looking at the panel, I know that you're going to get a steady dose of ophthalmology for the next 3 companies. So why is that so interesting? Well, I think most obviously it's a contained organ. And maybe what's not so obvious is that there is a huge unmet need. The traditional pharmaceutical ophthalmology companies had largely avoided the orphan space, and in fact, when we were first going out there and talking about orphan ophthalmology, no one even understood that there was such a thing, or had really even heard of these diseases, or knew of the millions of the patients that suffered from them. Also, what attracted us to orphan ophthalmology is that unlike other orphan areas where sometimes you struggle to find out what is that clinically meaningful endpoint, how do you measure it, how do you teach people to measure it, and how do you negotiate with the FDA, in ophthalmology, the endpoints were very well known. Visual acuity, visual fields, contrast sensitivity, and color vision, all well understood by the FDA, by the EMA, and by clinicians. So we thought that was a big advantage in the ophthalmology space. So I'm going to click really quickly through these slides, and I don't know if I can go backwards, and sit on the pipeline slide. So we have several programs in the pipeline: X-linked retinoschisis, about patient population about 35,000, achromatopsia, patient population of about 28,000, and XLRP, or a patient population of about 20,000. We recently added, through financing provided by our follow-on financing, a program in wet AMD. And what we're doing is kind of taking a step back, looking at the advances that are being made within the AMD space, understanding the multiple targets, the advances that have been made in AAV, and how to deliver and how to select capsids, and really putting forward a next generation program in that space. And now I'm going to kind of click through this because we can cover this in questions, and people can find this presentation on our website, because what I want to sit on before Josh and I start talking is the milestones for the year. This is going to be a huge year for AGTC, multiple INDs being filed over the next 12 months, and multiple data points rolling in. Initially, it's going to be the XLRS program. We'll start seeing data from that this summer. Achromatopsia, we'll start seeing data from that in the fall. The achromatopsia follow-on program, which is the second gene that causes achromatopsia, we'll have data for that in 2016, and then entering the clinic with XLRP, as well as entering the clinic with our wet AMD next generation program. So lots of data, lots of programs, an experienced team of people, and I look forward to talking to Josh about it.
Great. Well, this is a perfect slide to dwell on, I guess, with the initial trial for XLRS. Maybe just flesh that out for us. How many patients are going to be enrolled, the kind of patients who you'll be evaluating, and what is it that you're looking to see?
So it is a phase 1/2 study, which is traditional in orphan drug development. We're going to enroll 29 patients is the current plan. It is a dose escalation study in the first part of the program, and that will dose range through 3 different doses, all of which, based on the animal data, have a potential to be efficacious, and all of which were safe in primate studies. Then we will find the maximum tolerated dose and enroll another set of patients that will include children down to the age of 6. 6 is about the youngest that the retinal specialists feel that the kids can kind of reliably and patiently accomplish all of the testing points. So that's the plan for that trial. There'll be 4 sites across the United States.
And so what of the data will we see by the end of the year?
So by the end of the year, we would hope to have the initial data points for the full set of patients. The testing in the beginning of the trial is very frequent, day 0, day 1, day 7, day 14, 1 month, etc. So there's quite many test points. No one should expect that we will release the data patient by patient or test point by test point. We'd like to bundle up a cohort of that patient data before we move forward. We are testing for safety as well as visual acuity, visual fields, and OCT measurements.
Maybe you can step back and tell us a little bit about XLRS. What is the gene defect in morphology, and how does that inform your choice of vector selection and delivery modality?
Right. So XLRS and achromatopsia are both similar indications in that they have clinical phenotypes that are stable over time. So these are not indications where someone has a certain health of the eye, a certain vision, and then it gets worse and worse and worse over time. These are stable indications, which is why we selected them as our 2 lead programs. So they have that in common. What's different about the 2 indications is that XLRS is an indication where you're missing a structural protein. And that structural protein in the back of your eye holds the layers of your retina together. And that's very important because if the layers of your retina, which are highly specialized cells, can't talk to each other, then the electrical signal that turns photons into electrical signals and then into vision in your visual cortex, that electrical signal can't get back. And so that's why these people lose vision. So in XLRS, they have these delaminations in the back of their eye, and we thought that in this specific indication that subretinal injections were really just not the best path forward. So we're moving forward with an intravitreal injection in this specific indication. And that was important to the clinicians as being the safest path forward, but it does pose challenges because in primates, primate eyes are highly specialized, and unlike lower mammals, they have a macula, a fovea, an inner limiting membrane, and to get the AAV to go all the way to the back of the eye where you really want it to be, in this disease, it's a disease mostly of the macula and fovea, so we wanted to make sure we selected a vector that could get to those areas. And so we did our screening in primates. We think that's very, very important in ophthalmology gene therapy, that the screening you do for promoters, for regulators, for capsids, for delivery methods is done in a primate model, just because primate eyes are that different from lower mammal eyes. So we did our screening in primates, selected the one to move forward, and now, as we announced several weeks ago, we filed the IND.
And what are the visual complaints or visual loss severity typical of these patients, and what kind of gains are you realistically hoping to achieve? What's kind of that minimum threshold where you say, hey, you know, we're having an impact here?
Right. So these patients have vision between 20/120 to 20/60 at the very high end, and just to kind of bench that for folks, to get a driver's license, you have to have best corrected visual acuity of 20/40. And remember that these people can't improve their vision by putting on a pair of glasses. They don't have refractive errors in their cornea. It's not something you can wear a pair of eyeglasses for and correct. And so what we expect is, you know, the gold standard is that you're supposed to be able, Wiley Chambers has said, you're supposed to be able to improve vision by those 3 lines on the EDRS chart. And that would be our ultimate goal. In a phase 1/2 trial, where you're looking for what is the potency in humans and what is the right dose range, what we're looking for is improvements in the schisis, or that delamination, and improvements in visual acuity.
Do you need to improve all patients, or is there, if you get a fraction of patients responding, how do you think about the utility of that product?
Well, I think in a dose escalation orphan gene therapy trial where you're only dosing 3 patients in each cohort, I don't think we're going to get statistical significance. That's not the point of this trial. That is what we would try to accomplish in a phase 3, a pivotal trial where we've selected the dose and we're having a greater number of patients at that dose. So I don't think we need to see improvement in every single patient to feel comfortable about moving forward.
Okay. And maybe you can tell us about the achromatopsia program and how that program is optimized for the disease indication.
Right. So achromatopsia, while it's similar in that it's a stable phenotype, is a completely different disease. In achromatopsia, what's missing is a protein that's part of that phototransduction cascade in the membranes of photoreceptors, specifically cone photoreceptors. So in this disease, that phototransduction cascade does not occur because of the missing protein. So the photon hits that cone photoreceptor and nothing happens. But what we do know in achromatopsia patients, by working with very sophisticated adaptive optics that was actually adopted from the Hubble telescope, is that these patients do have live cones in the back of their eye. And so in this disease, this is not a secreted protein, so it's not in the outside of the cells like in XLRS. So we have to get to as many cones as possible. So we are doing a subretinal injection because these patients actually have very healthy eyes. So none of the surgeons have an issue with doing subretinal injections. And we screened, again, in primates, for promoters and capsids that we thought would give us the best chance of transducing every single cell type that are cones. Because an important issue with achromatopsia is that the animal models, while they're naturally occurring and very powerful, and we showed effect in the animal models, animals only have 2 types of cones. Lower mammals only have 2 types of cones. Primates have 3. So we wanted to make sure that we had promoters and capsids in a primate eye that got all 3 of those cone types. So again, we did that important screening process, and we're able to show we could light up all of those cones. And so we'll be going forward into the clinic with a subretinal injection with a proprietary vector, a proprietary promoter that we have shown can transduce all of those cone types. And importantly, these patients, achromatopsia, they don't have cone function, and people kind of think of it as being color vision, but achromatopsia has absolutely none of their cones work. So not only can they not see color, they are also legally blind. They have vision of 2200 or less, which means they can barely make out the big E on the eye chart. That is the severity of their vision.
Maybe we'll see if there's a quick question in the audience. And I think if not, we'll get ready for our next talk. A big year in ophthalmology, and for you guys, be a very exciting year, and look forward to the updates.
Great. Thank you, Josh.
Thank you, Sue.